Okay, so I'd like to start by thanking you all for being here and for supporting the laboratory and coming to listen to some of the research that we do here at the lab, and it's a pleasure to be able to be here tonight and talk to you about it. I like this slide. My eldest daughter helped design this when she was a little tot a long time ago. And this is what we want to talk about: how cells communicate with one another, and how cells communicate with their environment, and how changes in the environment are registered by cells and lead to changes in the way they grow, they divide, they survive, they move, they metabolize food, etc. We also want to talk about, to illustrate how those changes, how those mechanisms are altered in disease, and how that gives us new opportunities for designing new therapies to attack major human diseases. So the general area I'm going to talk about is called signal transduction. And that is just studying the mechanism by which the cells recognize a signal and translate it into an intracellular response, a physiological response. And a critical aspect of this is a process called protein phosphorylation. And that is the addition and removal of phosphate from proteins. So in response to a stimulus, you have a class of enzymes called kinases, which put phosphate groups onto proteins, and phosphatases, which take the phosphate groups back off again. And it's how these enzymes are coordinated that controls the response process. And my lab is interested particularly in the family of protein phosphatases. And I'm going to talk to you primarily tonight about an enzyme I discovered 30 years ago. It's making me feel old just thinking about it. But 30 years ago, and to illustrate how basic research into studying its function and its mechanism of action is suggesting new ways and new ways to treat diseases, as Sydney said, such as diabetes and obesity, breast cancer, and most recently, an autism spectrum disorder called Rett syndrome. Okay, so we're interested in how cells respond to changes in their environment, and that includes the response to hormones, to growth factors, to cytokines, etc. And so we're looking at the way cells respond to endocrine signaling, how a hormone, such as insulin, is produced in one part of the body, in the pancreas, and then is transported through the blood to a distant target cell in a different tissue, such as the muscle or liver or fat. We're interested in paracrine signaling, how one cell can secrete a protein signal that is registered by a target cell, or actually is registered by itself and controls its own function. And then there are also mechanisms by which cells can sense one another and touch one another. And actually these controls are very important in exercising control over cell proliferation and preventing cells from proliferating aberrantly. So this is all about how we sense these changes and these signals in the environment. And so what happens is that all cells possess receptors on their cell surface. These are proteins on the cell surface that can recognize specifically these target molecules, these stimuli, these hormones or cytokines or even cellular stresses. And the signal transduction process that we're talking about is how the receptor can now set up what we call a signaling cascade, a set of interactions between proteins that culminate in a change in the nucleus that leads to gene expression changes. You have certain genes that are switched on, certain genes that are switched off, and this results in a functional response. Now, the critical part of this that we focus on is early on in the pathway, and what happens when the stimulus and the receptor come together, and how this signaling cascade, as we call it, is initiated to set up the changes in gene expression that underlie the functional response. And this is the process of phosphorylation. And so Phosphorylation is the addition of phosphate onto a target protein. And so this is a summary of what happens when a receptor on the surface of the cell registers one of these stimuli, a cytokine. And so these are proteins on the surface, so this is the cell membrane. There's a portion of these proteins on the outside of the cell facing into the extracellular space, and there's a portion on the inside that's responsible for the signal. So the bit on the outside can recognize specifically the stimulus, such as the cytokine, and then on the inside of the cell, there are enzymes called kinases that transfer phosphate onto proteins, phosphate onto the receptors itself, that lead to interactions between the receptor and other proteins that set up the signaling reaction that is sent to the nucleus. And I'm trying to think of an analogy for how to describe phosphorylation, and I got a beautiful one that I really like. This discovery of phosphorylation was made in the 50s by Eddie Fisher and Ed Krebs, and it has just been fundamentally important for those of us working in this field. And Eddie and Ed won the Nobel Prize in 1992 for this discovery. And when he was introducing the field, Hans Jornvall came up with the following analogy. And I like it because both of my daughters were ballet dancers. And this is my youngest daughter, Caroline, and she's about to embark on a career as a professional ballerina. But Hans Jornvall's analogy was phosphorylation is like putting on ballet shoes. When you put on the ballet shoes, the dancers come together and absolutely amazing things happen on the stage. Phosphorylation is putting on phosphate onto proteins. Proteins come together in amazing ways and wonderful things happen within the cell. So overall, we're looking at, again, the ligand binds to its receptor, phosphorylation occurs, and then these proteins now interact with one another. The messages are sent down into the cell to give you the physiological response, changes in gene expression, and then the pathway is switched back off again because the phosphate is taken off, and now the pathway is switched off. So you want to be able to control these pathways. So when you have a response to a stimulus, you want it to be a transient response. You don't want to continue growing all the time. You don't want to continue metabolizing all the time. You need to be able to control the responses. So putting the phosphate on is important, and taking it off is also important. And if you don't get that timing right, you disrupt these normal signaling pathways, and that can lead to disease. And by looking in cells as to how these signaling pathways are disrupted, that's where the potential for therapeutic intervention comes in. And that's what I want to illustrate. So when I started out in this field, after I'd done my PhD in Dundee, I decided at Oxford and at Dundee, it was phosphorylation that interested me. This aspect of biochemistry was what I was very excited about. But I was also sort of a contrary type of person. And everybody was interested in the kinases, the enzymes that put phosphate onto proteins. So I decided to look at it from a different perspective, the phosphatases that take it back off again. And it was interesting because this was in the mid-'80s. And we knew that, first of all, that phosphate is added really to 3 different amino acids in proteins. There's 20 different amino acids that make up proteins, and 3 of them can accept phosphate: serine, threonine, and tyrosine. And it was at this time that there was a lot of excitement about enzymes that put phosphate groups onto tyrosine residues in proteins, because they had been established as the products of oncogenes, proteins that cause cancer. And they had been established as the products of receptors for growth factors that promote cell growth. So there was a lot of excitement about these tyrosine kinases. And they were just starting to understand about how the signal transduction pathways were put together. Really, it was a few years after that, it was discovered that these phosphorylated residues are recognized by reader proteins, and that when these proteins form complexes together, that's what transmits the signal. But these fellows here, called the erasers, I didn't think that was very good either, because it suggests that they're just housekeeping enzymes, but they're much more important than that. But nothing was really known about these enzymes. And so that's what I set out to do when I joined Eddie Fisher's lab at the University of Washington in Seattle in 1985. And so in those days, this is ancient history. This is, cloning was really only just becoming established, and it was before the Human Genome Project. And so it was a very interesting time for research. One of the things that we had to do to be able to isolate one of these proteins was to have a way of measuring its activity. We had to generate a protein that we could use to measure the activity of the phosphatase we were trying to purify. And I won't go into the details, but I spent the first 4 months of my postdoc coming up with this. And it was it's a protein that you can buy commercially called lysozyme, that we denatured and chemically modified so that it was now soluble, and found a way to phosphorylate it on just this one site. And so now we had a protein that could measure phosphatase activity over a broad range of substrate concentrations. So that was a real advantage. That was very important for trying to identify the nature of the phosphatases that are important in these pathways. And so what I set out to do was to purify one of these enzymes. And so we did this from human tissue, human placenta. So there was lots of trips to hospitals to get tissues, and then lots of hours in a cold room trying to identify and isolate the protein. And we found a way, using this substrate, to come up with a modified form of the protein that is recognized by the phosphatase and can pull it out of a complex mixture of other proteins. And so what we did, this was important. What this shows is something called a polyacrylamide gel. It's a sieving mechanism that allows you to separate mixtures of proteins according to their size. And then you can stain with something, a blue dye called Kumasi blue, that allows you to visualize those proteins in the stain, in the gel. And so what has in panel A here, I've gone through 5 different steps of purification, which is a lot. And I was very pleased with myself, because when I looked at the gel, I thought, I've almost got the protein pure. All I've got to do is get rid of all these other things here. So I made this affinity column that should bind to the phosphatase specifically, and then looked at what didn't bind to it. And lo and behold, look at all this stuff that didn't bind. And it turned out that this thing here, this minor protein here in the mixture, was the purified enzyme. And this is what we were able to isolate. And so it was the power of this approach that allowed us to get this enzyme purified for the first time. And I can tell you, there's nothing more exciting than when you're in the lab and you have a tube of material and you think you're the only person in the world who's got it. It's quite a feeling. So what were we able to do? Well, a lot of science is actually being in the right place at the right time. And at the University of Washington, we were on the 3rd floor in Eddie Fisher's lab, but on the 4th floor was one of the experts in protein sequencing. So proteins are made up of amino acids, and the order of the amino acids determines the shape of the protein, the function of the protein. And so Ken Walsh's lab was one of the experts in determining the order of amino acids in proteins. And with Harry Charbonneau, who was a senior postdoc in Ken's lab, we got together and determined the sequence of the proteins in our phosphatase that we just identified, called PTP1B. And in those days, the number of proteins that we knew the complete sequence of was rather small. And when we put it into the literature, put it into a database of protein sequences, we got lucky. And that's the second element of what's important in science. It's really important to be lucky. And once you are lucky, you've got to make sure you harness that luck properly. And why we were lucky was there was a body of literature on a molecule called CD45. And this is a protein that defines immune cells in the blood system. It's a marker of immune cells, hematopoietic cells. And so what was important about this was it looks like one of these receptors that I just talked to you about that have kinase activity, except this receptor has phosphatase activity. And so this changed completely how we think about the control of signaling pathways. It's not just kinases triggering pathways by responding to stimuli in their environment, it's also phosphatases responding to those stimuli. And it's the coordinated and competing action of the kinases and the phosphatases that controls the outcome of the signaling pathway. And so Eddie and Harry and myself, we got invited to write a review for Science to describe this new discovery. And it was quite funny, actually, when we got the galley proofs back. We'd drawn up a table that summarized everything that was known about these enzymes at the time. And you can see that the copy editors at Science were also really excited about the work. It's meant to say organism. But so anyway, I hope you'll share the enthusiasm, but act with a little more restraint than the copy editors at Science. But the point is that when you're thinking about signaling, you have to look at both the phosphatases and the kinases. And it's how these two families of enzymes are integrated to control the phosphorylation of target substrates that regulates how cells respond to stimuli. So how can we exploit these enzymes therapeutically? So the first example we got into is something that is one of the major healthcare challenges that faces Western civilization at the present time. And it's the prevalence of obesity. So, some of these are staggering statistics, and I'm sure you see these in the paper all the time and see them on television. But it's estimated that the prevalence of overweight and obese people rose by 27.5% in adults, 47% in children. That's the scary number. In 2013, there were 2.1 billion people overweight and 671 million obese adults. The world prevalence doubled between 1980 and 2014. And as you can see, the problem with all of this, particularly the fact that it's affecting children, is that really obesity could mean that this next generation could be the first generation that won't live longer than its parents. This is a serious healthcare issue. And if you look in the United States, just in the United States, if you go back to the mid-80s, this is the color coding here is the percentage of obese adults. So that's a body mass index of greater than 30. And these colors are showing the increasing hot colors show increasing obesity. If you go back to 1985, these were all the very cold colors. Now it's changed, and you can see by 2005, there were many that were in the 25 to 30% range, increasing again, getting hotter colors by 2010, and you can see now in 2014, some cases more than 35% of the population in some states is obese, but in overall, greater than 30% of adults in the US are obese. So this is a huge problem. We're thinking about obesity and we're thinking about insulin signaling. So this is an example of one of these receptors that's on the surface of the cell that registers a hormone, insulin. Binding of insulin to the receptor then causes changes in phosphorylation that triggers, it doesn't matter the details of this, but it triggers this signal transduction pathway that is involved in controlling glucose homeostasis, the levels of glucose in the bloodstream, and the disposition of glucose into other tissues. And so the problem when you become obese is that you become insulin resistant. So insulin circulating in the bloodstream would normally bind to its receptor and trigger this signaling pathway that will regulate the levels of glucose in the blood. But as you become obese, you become resistant to the effects of insulin. And then ultimately, the pancreas can't produce enough insulin to give you a response, and that's when you become diabetic. So what we're looking for are ways to think about how to treat this approach. And what was discovered was that this enzyme that I purified 30 years ago, PTP1B, Michel Tremblay's lab and Ben Neal's lab generated knockout mice where they removed the gene from the mice and looked at what happened to the phenotype, what happened to the properties of the mice. And they found a dramatic phenotype. This is a wild-type mouse fed a high-fat diet, the equivalent of hamburgers and French fries. This is a PTP1B knockout mouse fed exactly the same diet. Astonishingly, The PTP1B mice display enhanced sensitivity to insulin, and the mice are resistant to the development of obesity and type 2 diabetes when fed the high-fat diet. So what this suggests is that if you can come up with a drug that will inhibit PTP1B, you may have a way of targeting obesity and diabetes. Insulin resistance, which is diabetes, and obesity, which is leptin resistance, both of which can be targeted by an inhibitor of PTP1B. So many people set out to address this. I was involved with a company that was doing this, and so the standard approach that you take is that you would look for small molecules that will bind at the active site of the enzyme and inhibit it. And so what you tend to do is you will take an active enzyme, this is PTP1B, and you'll find a way to measure its activity, or you'll find a way to measure the binding of small molecules to the protein. And then you test the effects of hugely differing arrays of small molecules. Typically, drug companies will test 1 million compounds, and some of the latest DNA library encoded technologies, they're testing billions of compounds against individual targets. But what you look for is a small molecule from one of these screens that will bind at the active site and will inhibit the activity of the enzyme. And then what you do is you go through modification, you you apply chemistry, medicinal chemistry to this to modify the structure of the compound so that it now binds to your target with higher affinity and greater specificity and creates a small molecule drug that can target that enzyme in cell and animal models and also in patients. And we came up with one such molecule. And so this was a potent inhibitor of PTP1B. It was a reversible inhibitor of PTP1B. It was a selective inhibitor of PTP1B. It had all the properties that we would want. But its structure was highly charged, and so it was limited in what we call oral bioavailability. In order to take it, you had to inject it. You couldn't swallow it. And so here we are with a fantastic scientific breakthrough where we've generated a small molecule inhibitor of this important target, but industry isn't interested because they don't want an injectable, they want an orally bioavailable drug as the next generation of drugs to target diabetes and obesity. And that was where the undruggable bit came in. They'd taken this standard approach that is always used in industry, where you target the active site of the enzyme, but the chemistry of the compound and the chemistry of the molecule, of the protein, and the chemistry of catalysis and the features of the active site are such that the best inhibitors do not allow them to be, the properties of the active site show that the best inhibitors are highly charged and therefore not orally bioavailable. So it was decided by industry that these are not druggable. You can't make drugs that'll target this family of enzymes. So when you're in an academic setting, that's a bit like a red rag to a bull. That makes you think, I discovered this family of enzymes and I was really ticked off by this. There's got to be different ways of thinking about how you can approach this problem. And that's the importance of academia. You have the freedom to think differently and to think broadly about a difficult problem. And so what we did was we decided, okay, well, let's move and look for different ways to inhibit the enzyme. And let's look at different diseases. Diseases for which oral bioavailability, having to take the drug as a pill, is not such a problem. And so we moved to cancer. And so there's been a lot of progress made over the years in targeting signal transduction pathways in cancer. So the cancer condition is associated with alteration in signal transduction pathways that leads to cells proliferating aberrantly, to surviving aberrantly. And so what we're trying to do is looking at the features of those signal transduction pathways and seeing if we can target those features that are unique to the cancer cell. Can we target those with small molecule drugs? And so there's a couple of examples just to highlight that have been spectacularly successful, but have also illustrated some of the limitations of the approach. So in the first one, many of you will have heard of Gleevec as a small molecule inhibitor of a kinase called BCR-ABL. And this is a kinase that is associated with a particular form of leukemia called chronic myelogenous leukemia. And it's caused by a chromosomal translocation, which means that normally you have chromosome 9 and chromosome 22 as separate entities, and you have a gene on chromosome 9 called c-Abl, and a gene on chromosome 22 called BCR. But in this particular form of cancer, these two chromosomes fuse, and what you end up with is a fusion protein that's produced as a consequence of this Philadelphia chromosome, this aberrant chromosome that is a unique defining feature of chronic myelogenous leukemia cells. And so what you end up with is a kinase where you've lost bits out of the kinase and replaced them with bits of the BCR protein. So you've created a unique protein that is specific, a marker for the cancer cell. And so what Gleevec is an inhibitor that targets this unique feature specifically. And it had spectacular results. But also what happened was that the cancer cells have genomic instability, they can change. Eventually, the patients develop resistance to the therapy. And this is a common theme amongst drugs that target kinases in a cancer context. The other one I want to mention briefly is a different way of approaching, but it's again a specific marker of a cancer cell in the context of breast cancer. So in this case, it involves a family of these receptor-like molecules called the HER family, and HER2 is one of these, and HER2 is a marker of HER2-positive breast cancer cells. So there's 4 different HER family members, and what they tend to do is come together in partnerships where driven by a ligand that sets up these signaling pathways that are associated with proliferation, motility, invasiveness, etc. In breast cancer cells, the level of expression of HER2, certain breast cancers, the level of expression of HER2 changes dramatically. So instead of having a few thousand copies of the cells, you have a few million copies of the protein on the surface of the cell. But that gives you a specific marker that you can target uniquely on the cancer cell. And so people have been taking a variety of different approaches to doing this. There are kinase inhibitors that target the kinase domain of the HER family proteins and inhibit that. But also there are antibodies, proteins that recognize selectively the extracellular segment of these molecules. And this one, trastuzumab, is also known as Herceptin. And it's able to target the extracellular segment of HER2 specifically. And this targeting prevents, it does a number of things. It sensitizes the tumor to an immune response, but it also disrupts signaling. It prevents the dimers from forming and the signaling pathway from being initiated. Another version of this is to use the antibody to bring a toxin specifically to the cancer cell, and this toxin is then involved in killing the cancer cell specifically. Again, these have had spectacular results, but with limitations. Only about 30% of patients that have this amplification of the HER2 gene respond to Herceptin, and those that do respond tend to become resistant within about 18 months. So again, what we need are new approaches, new targets, new strategies that we can use in conjunction with drugs like Herceptin and Gleevec, or on their own to try to think about how new ways to target these cancer cells. And so do the phosphatases offer such a strategy? And the answer is yes. It turns out that this enzyme, PTP1B, is in a gene locus that is frequently amplified in breast cancer, and that overexpression of PTP1B alone is sufficient to cause breast tumors. And in these mouse models, what was found was that if you take a mouse model that expresses this HER2 oncoprotein and cross it onto another mouse background that lacks this phosphatase PTP1B, the incidence of tumorigenesis is decreased, and the metastasis, which is what kills you, is also decreased. So this says that this phosphatase, which functions to switch off insulin signaling and switch off leptin signaling in diabetes and obesity, functions to switch on signaling through HER2. And that an inhibitor of this phosphatase could be a new way of thinking about how to target HER2-positive breast cancer. So we have a new approach. So we have a new disease to look at, HER2-positive breast cancer. We wanted a new approach. And so the approach we took, we knew an awful lot about the structure of this molecule PTP1B that I purified, and we know a lot about how it works. But the version that we'd all worked on is shown as this oval here. It's an isolated domain. But it's a fragment of the way the protein is normally expressed in the cell, and normally it's targeted to membranes through a region here that serves a regulatory function. And so, to cut a long story short, what I wanted to do was to look for small molecule drugs that would target this long form of PTP1B specifically compared to the short form that everybody else had been using in their drug screens. And so what we found was such a molecule. It's a natural product called traduscamine, or MSI 1436. And it binds here in this regulatory C tail, and it binds here also on the opposite side of the enzyme to the active site. And what happens is that PTP1B is a phosphatase. The active site is open before substrate binds. When substrate binds, the active site closes around the substrate, and it adopts a form that now allows it to function as a catalyst, now allows it to dephosphorylate the substrate. In the presence of the inhibitor, it's locked in the open conformation. It can't close around the substrate, and it can't catalyze dephosphorylation. So we have a new way of inhibiting this molecule that targets parts of the protein that are distinct from the active site. So we were hugely excited about this. So we tested this in, when I say we, this is the work of Navakrishnan, a research associate in the lab. We tested this in various cell and animal models of HER2-positive breast cancer. And this is just one piece of data to show you. This is a mouse model that expresses an activated form of HER2 and develops a breast cancer like the disease that's encountered in humans. And what you find is there's metastasis to the lung. So this shows sections through the lung of the control mice, and you can see these horrible metastatic lesions. This is what ultimately results in the fatality. But when we treat the animals with the inhibitor at 2 different doses, you can see that the metastasis has essentially been wiped out. It's a very, very dramatic result. Now, this molecule had already been in patients, in obese patients, as part of a previous clinical trial, and was extremely well tolerated. And so these data combined with that result, we've now started a clinical trial in collaboration with scientists at Northwell Health, formerly North Shore LIJ. The FDA has asked us to repeat a phase 1 study. So we are now recruiting patients. The patient that we've already injected the first patients. It's an incredibly exciting time. So the process of recruitment has started. The clinical trial has started, and we're waiting with bated breath to see what happens. But it's an early part of the pathway. For those of you who don't know the various phases, there's a preclinical phase to drug development where you look in animals to see how the animal interacts with the compound, how the compound interacts with the animal, looking for toxicity effects, etc. If it passes that, which this has, you then go into a phase 1 clinical trial, which is designed to assess the safety, the tolerability, again, how the compound interacts with the patient and the patient with the compound. And if you get through that, which we anticipate, we're hoping for good results in this, then it starts to get longer and more expensive. You then have a phase 2 trial where you assess how well the drug actually works, where you're looking at dosing and efficacy to see to what extent it can attenuate tumorigenesis, attenuate metastasis. And then if it passes that, you have a phase 3 trial, which is a huge trial where you have to show that this new therapy is better than the gold standard treatment that exists. And so what we're hoping is that actually, this approach for which Herceptin is the standard of care, we're hoping that this approach may be in conjunction with Herceptin, may be able to extend the time before you encounter resistance, or even this approach may be able to address patients who have become resistant to Herceptin. And that's what we're pursuing at the present time. We've also come up with analogues of this molecule that are orally bioavailable. So what this shows you is a dose response with different amounts of this analog. And this is the control animal fed the high-fat diet, and this is the animal fed the analog of 1436. And the importance is that it's fed the analog orally, by oral gavage. So we have this inhibitor now of PTP1B that is also orally bioavailable. And we're hoping that this will re-engage industry to think again about diabetes and obesity. And also, these are natural products, so they're very difficult molecules to work with. What we have gone on and worked in collaboration with scientists at the Rockefeller University, who have a library of 180,000 compounds, and we've screened their library and come up with 28 compounds that now show similar properties. They prefer the long form, inhibition of the long form of PTP1B over the short form, so they function in the same way as these natural products like tridoscamine, but they are chemically simpler, easier to work with. And so while the clinical trial is ongoing with 1436, tridoscamine, we're also now developing these new analogs as a way of coming up with next-in-class inhibitors and hope to actually improve upon results we may get with 1436. Okay, so for the last few minutes, I just want to switch gear and just to illustrate how the basic research that we've been doing has taken us into really unexpected areas. And actually, this is a testament to working in a place like Cold Spring Harbor. There's a lot of different kinds of research that goes on here. And when you go to seminars, you hear things that you would never hear anywhere else. But it also sets off ideas in your mind that you wouldn't get if you weren't in a place like this. And this is an example of the research associate in the lab, Navakrishnan, going to a seminar by another postdoc who was working on a mouse model for a disease called Rett syndrome. And I'll never forget, he'd come running back after the seminar saying, "What do you think of this?" The phenotype that they were reporting suggested to him there may be a role for PTP1B also in this disease. So let's just spend the last few minutes talking about that. So this disease is a combination of symptoms of autism, cerebral palsy, Parkinson's, epilepsy, and anxiety disorder. It's a disease called Rett syndrome, and it's an extremely distressing disease because the kids are born normally, but then after 18 months or so, they start to develop the symptoms. And the symptoms include, it originally looks like an autism spectrum disorder, but then that goes away, and it's replaced by cognitive, motor, respiratory, GI symptoms. There's increased risk of seizures. The kids can't walk properly. The kids can't talk. They can't communicate. So it's a very distressing disease. It predominantly hits girls, affects girls. They live into adulthood. They require total 24-hour-a-day care, and there's currently no treatment for this disease. As I said, it places an enormous burden on the families. The patients require 24-hour care. There's lots of hospitalizations and surgeries, need for special treatment, special education, home health aides, etc. It is a profoundly disabling disease. So it's caused by a change in a protein called MECP2. And MECP2 is one of these molecules that's a transcription factor that regulates gene expression changes. So 95% of cases of Rett syndrome are caused by loss of function of this MECP2 gene, this transcriptional regulatory protein, and that regulates the expression of a wide variety of proteins. It acts relatively late during development, and it's X-linked. So females that have two X chromosomes, they can afford to lose one, one copy of the gene, but males, which have one X and one Y, if the X-linked MECP2 gene is mutated, the males, it has a much more profound effect in males, they tend to die. And so most of the patients that develop are females. The loss of MECP2 function leads to decreases in gray and white matter, decreased size of neurons, but these effects are not irreversible. In fact, actually, a really important development came recently where animal models of the disease were developed in several labs. People like Adrian Bird, Rudy Yanish, Huda Zogby, they showed that you can develop mouse models where you disrupt the function of MECP2, and most importantly, they showed that if you can reintroduce MECP2 and switch it back on again, you can reverse the effects of the disease. So this suggested that this may be a disease that is addressable by the kind of approaches that we've been taking to target signal transduction pathways. So what's actually going on in the disease? What has been found is that loss of MECP2 results in global changes in neuronal function. Some parts of the brain, neurons are now hypoactive, they're less active. In other cases, they're hyperactive. There's changes in neurotransmitter levels, there's changes in proteins that regulate neuronal function. But attention has focused on one particular molecule, brain-derived neurotrophic factor, or BDNF. And this is just to show that BDNF is expressed in particular parts of the brain, in particular cells, but it's a fundamentally important molecule that controls dendritic and axon growth, synaptic maturation, synaptic plasticity, long-term cellular functions, and that it's known that deficiency phenotypes result in several of the symptoms that overlap with what you see in Rett syndrome. And BDNF is one of these things that interacts with a receptor molecule on the surface of the cell that is able to promote tyrosine phosphorylation dependent signaling pathways. And so the approaches that have been tested now are focusing on enhancing BDNF signaling and signaling through this its receptor on the surface of the cells, which is called TrkB. And these approaches include trying to increase the levels of endogenous BDNF, which are decreased in the disease, but also to improve the signaling of TrkB. And actually, this is where phosphatase inhibitors come in. And so this is the contribution that we've made in the lab, and by we I say particularly Navakrishnan. So what we noticed, what got us interested in this was listening to this talk where Nava noted that the phenotype of the mice expressed features that suggested there was too much PTP1B. It was the opposite to what you see in the PTP1B knockout mouse. And actually, when we tested, that's exactly what the situation is. So in the wild-type mice, this is samples from the forebrains of 8 separate mice. This is one of these STS-Page gels again. This is the level of MECP2, and this is the normal level of PTP1B. But when MECP2 is disrupted in the disease model, you can see the level of PTP1B is dramatically elevated. So that led us to postulate, well, if PTP1B is dramatically elevated, and we have these small molecule drug candidates that can target PTP1B, to what extent can these ameliorate some of the symptoms of Rett syndrome and offer us a new idea of how to attack this terrible disease? So the first thing we did was to look in the male mice. And as I said, the male mice tend to die. It's a much more difficult, in the females, they can compensate for the changes in one allele, but in the male mice, they can't. And you can see that if you treat with this inhibitor, the one that we're using in the breast cancer trials, you can see that the animals that are treated with saline alone only live for about 50 days, but we can extend their lifespan about 2-fold by treatment with the PTP1B inhibitors. That's already a dramatic result. So, but really what you want to understand is what's going on in the females. And so I'm trained as a protein biochemist, and this is taking us into animal models and looking at behavior, and this is very unfamiliar territory for me and something that's actually quite daunting. But there's an assay that we've been able to produce, reproduce, that even somebody like me can understand it. It's very simple. And so it involves, there's a classic hand movement, a dystonia, that's a phenotype in these patients. And this is mimicked in the mice. If you pick the mice up by the tail, the wild-type mice will do this with its paws, trying to grab onto something. The Rett syndrome mice will do this. So the assay is simple. You pick the mouse up by the tail and count how long it does this compared to do this. And what happens is, you can see that the wild-type mouse, the paw clasping is minimal. But in the MECP2 mutant mouse, you can see that for the most part, they clasp their paws together. But when you treat them with the PTP1B inhibitor, that paw clasping phenotype is essentially abrogated. Another impact of an inhibitor of PTP1B on this phenotype. So the last, I just wanted to show you some pictures because I think they're rather exciting. And it's a behavioral assay that was done, again, as part of a collaboration here with Steve Shea and Kirti Krishnan, who's another research associate who was working with Steve Shea to develop this assay. And it's a pup retrieval assay, and it compares the ability of the wild type and the mutant animals to retrieve pups. So you house the mice with the pregnant mother, and then you begin treating with the compound. And this results in about 10 days of treatment. And then once the pups are born, the wild type and the het mice receive training from the natural mother. And then you perform the assay that I'll show you on the wild type and the het separately. And you look at the time it takes them to retrieve all the pups. So, this first one, I'm going to go speed this up a bit. This first one shows you what the normal mother will do. So what happens is the pups are taken out, and you put the pups in the four corners of the cage, and you can see the mother retrieving the pups and bringing them back to a nest that she's created in the corner of the cage. So very quickly, the mother goes out, gets the pups, and brings them back in. Okay. Now, what happens if you have an MECP2 mutant animal, the Rett syndrome animal? This mother essentially ignores the pups altogether, just tramples all over them. And you can see that any movement is towards the nest is because they're being disturbed as the mother is running aimlessly around the cage. Okay, so you get the idea. Okay, now let's treat the animal with the PTP1B inhibitor and see what happens. The mother now more effectively picks the pups up and brings them back over to the nest. So another impact of inhibiting PTP1B on a behavioral readout in this mouse model. Okay, so in summary, this is another example of modifying a signal transduction pathway to have a therapeutic benefit. Under normal conditions, brain-derived neurotrophic factor sends a signal that activates the signaling pathway, leads to changes in gene expression that give you the normal developmental response. And in the presence of MECP2, PTP1B is normally suppressed. However, in the Rett syndrome model, where MECP2 is disrupted, the levels of PTP1B are dramatically elevated, and now it favors the inactive form of TrkB, which doesn't signal. And so this suggests that if we can inhibit PTP1B, we can now favor the equilibrium, switch it back to this form of TrkB that can now signal normally. So what I think we've shown is that this is a new approach to therapeutic intervention in Rett syndrome with small molecule PTP inhibitors that target classic signaling pathways. It's trying to approach this disease the way we thought about approaching diabetes and obesity, the way we thought about approaching cancer. But really quite exciting, it has implications for other autism spectrum disorders because obesity and metabolic dysregulation, which are features of Rett syndrome, have also been implicated here. So it'll be interesting to test in these models too. And also MECP2 has been implicated in other neuropsychiatric disorders, such as schizophrenia. So perhaps there's an opportunity also to test in those contexts. So, I mean, it's really exciting. This could be the start of looking at PTP1B in a variety of different contexts. But overall, it's reinvigorated, I think, these studies, PTP1B as a therapeutic target. The industry thought of this as non-druggable, but what we've been able to do is open up other major human diseases as well. And also, PTP1B is one of 100 members of a family of enzymes. There's implications for other members of the family that may give you different ways of thinking about other major diseases. So again, the take-home message is thinking about signal transduction not just from the perspective of the kinases, but also the phosphatases. And so, whereas up until now the phosphatases have been underexploited from the development of from the perspective of developing therapeutics, I think now we have a new avenue from which to pursue different new, unique approaches to major diseases. And so the take-home message, I really want to ram this one home. Basic research has a crucial role to play in developing therapeutics. Please support basic research. We need to be doing the kind of research that is just interest-driven. You know, there are, who would have known that people studying the color of petunia flowers would come up with a process called RNA interference, which is now used to validate all major drug targets in major diseases? Who would know that studying fluorescent proteins in jellyfish would give us approaches to studying signal transduction? Both of those things ended up with the Nobel Prize. We're not smart enough to know how everything we should be doing. We need to be supporting basic research. And the study of basic research will provide new opportunities. We've developed new ways to manipulate this highly validated target that industry viewed as undruggable by being in an academic setting, by using basic research approaches and thinking about new and different ways to target it. And in this current economic climate, there's going to be more of an important role for academia in this discovery phase of drug discovery. We're not going to be able to take compounds all the way to phase 3 clinical trials. It's just too expensive. But if we can come up with new and unique approaches and validate them sufficiently, we can hopefully re-engage industry and provide an engine for generating new approaches to treating major diseases. And I think in the future, research into mechanisms of signal transduction, particularly from a phosphatase perspective, will be new and fertile ground to identify new drug targets in cancer and major diseases. So our clinical trial is just starting. Hopefully there's lots more to come, and there'll be great excitement over the next few years. So I just want to stop here and thank some people in particular. So I started my research in this area as a postdoc in Eddie Fisher's lab at the University of Washington in Seattle. Eddie is one of the great guys of science. He was trained in the Geneva Conservatory of Music and had to choose as a career between a concert pianist and a scientist, so he chose science and won the Nobel Prize. He's sickening. But you can trace back the genesis of this entire field to the work that Eddie did in collaboration with Ed Krebs in the mid-50s, around the time Jim was working on the structure of DNA. And this is a picture taken with Eddie 2 weeks ago at a conference in Europe. Eddie is now 96. He attended every talk, and he had some really nasty questions for people at the end as well. So he's firing on all cylinders, and he's a personal inspiration. He's an amazing man. And this idea that scientists work on their own and they're sort of squirreled away doing things all on their own, that is simply not true anymore. We work as teams. And I've been very fortunate to have some fantastic people work in my lab over the last 25 years. It's been a privilege to work with them. And this is a picture of my group at the moment. Some of them are at the back here, and I hope they're not going to ask difficult questions at the end of the talk. But I'd particularly like to highlight Navakrishnan, who has really been responsible for all of the small molecule work that we have done over the last 2 or 3 years on developing inhibitors of PTP1B. Nava is a fabulous research associate, and I think he has a stellar career ahead of him. I'd also like to thank Dan Budman, Ruby Sharma, and Xinhua Zhu from the Monterey Cancer Center at Northwell Health. Dan is leading the clinical trial that we're doing, and it's a collaboration also with Depimed, which is a company that's set up to support that. And then the work we're doing on Rett syndrome is a collaboration with David Katz from Case Western, and it's a venture that's been set up called Rett Neurosciences that is a relationship with Monica Conrads, who leads the Rett Syndrome Research Trust, and is helping to accelerate the transition of ideas from academic research into the clinic. And then finally, I'd like to acknowledge the support. We do get grant support from the NIH, but there's a lot of people, local supporters of cancer research, that I am extremely grateful to all of you for your support. The situation for funding research at the NIH now is pretty dire. It's hard to get funded things that are exciting or cutting edge. I wrote several grants to support some of the research that ended up in the clinic with the PTP1B inhibitor, and it wasn't funded by the NIH. And if it wasn't for the support of these people, you know who you are, the local community, the work wouldn't have got done. So thank you very much, and please, we hope you will continue to support the research and the work that's done here at Cold Spring Harbor. Thanks very much.